Pierre Barennes, are you here? I see that you already shared your screen. Now you just have to unmute.
You see my screen?
Yes, we see, and now we also hear you.
Yes.
And please, the floor is yours.
Thank you. Okay, so thank you for the invitation. So I'm Pierre Barennes, PhD student in David Klasman Laboratory, and I will present TCR repertoire as a biomarker of immune disease with two examples. One case in COVID-19 and in autoimmune disease. So a brief introduction about why TCR repertoire could be an interesting biomarker. As you know, so T cells have a central role in the adaptive immune response involved in all immune disease like cancer, infectious disease, and autoimmunity. And the T cell express on their cell surface, so the TCR, T cell receptors, that is a hallmark of the T cell specificity needed to the activation. And the collection of all of these TCR expressed on the T cell surface represents the repertoire that reflects the immune history of an individual and has a predictive character on a future immune response. So that's why TCR repertoire could be an interesting biomarker. So a biomarker is a biological measure found in blood or other fluid tissue that's in a normal or abnormal conditions. And to find this biomarker, you need to design your strategy. First, to define your clinical question, to select the appropriate patient and control, and the two other parts that are interesting for the TCR repertoire analysis, as the selection of the analytical platform and the biological material, and also the statistical study. So, and for this part, you have, and very briefly, you have to design your experiment to know and to choose if you want to analyze T cell from total T cell or sorted T cells from tissue, organ, or blood, DNA, and RNA, and you could read this review to have more information about that. You have also a part of data production and to select the appropriate method to answer to your question, and also with a huge analytical tools, you have to select the appropriate analysis workflow. And here we try to create a new analysis workflow, and for that we validate first the workflow by try to prove that the TCR could be a prognostic biomarker or severe COVID. For that, so the question is, TCR repertoire could be a prognostic biomarker to evaluate the susceptibility to develop severe infection of COVID-19. For that, so we select from the huge cohort of adaptive biotechnology, we select early sample from infected patients with a known prognostic. So we select 40 patients died due to the infection and 40 patients that recovered. The repertoire came from total T cell from blood. We have only the beta chain, and both groups are comparable in terms of age and of day from diagnostic to sample. So we apply our feature selection strategy that I will not present here, is not yet published, but we split our 75% training data set. We collect signatures, so signatures that are enriched and those that are impoverished in the disease. And we try to classify the 25 the remaining data set. And here, this is just two examples of a prediction result in the confusion matrix. And we observe that with the signature, we are able to classify with a good accuracy patients that died from the infections compared with those that recovered from the infection. And to control this signature, we also try with a random signature composed by randomly selected CDR3. And we observe that here we don't have a good clustering separation, sorry. And we perform other iteration to validate the strategy, and we observe that with all signature, we are able to classify with a high accuracy the deaf patient and the recovered patient. So this is a proof of concept that the TCR repertoire is a reliable prognostic marker for COVID-19 infection, and this results validate our strategy. So we know that the TCR repertoire on total T cell is an interesting biomarker. So for the infectious disease that I just presented, and for the autoimmune disease, like the publication of flu 3 years ago, that they are able to classify lupus patient and rheumatoid arthritis patient compared to healthy volunteers. But, and as you know, especially in the autoimmune disease, due to the antinomic role of the regulatory T cell and the effector T cell and the immune balance between these two cell populations, we know that we need to know which cell population is behind this biomarker. First, to better understand this immune disease and to determine the biological role of our biomarker. And with all of this information, it could, in the next step, could be an interesting therapeutic clone. So in the laboratory, we collect a sample from healthy volunteers, from type 1 diabetes, from rheumatoid arthritis patients. We collect blood for each of these patients, and we sort the effector T cells and the regulatory T cells. We process to the RNA extraction, TCR library amplification, and sequencing. And here also we process to the feature selection in the, for 75% training data set. We collect the enriched signatures, so we always compare with healthy volunteers. The enriched signature are CDR3 enriched in the disease, and the impoverished signature are impoverished in the disease, so enriched in the healthy volunteers. And we try to classify the remaining test data set. So we perform a complete and important benchmarking by comparing different type of learner, but also different parameter on the prediction. And this analysis is still in progress, and we will evaluate the impact of each chain, each cell subset, and each signature. For this presentation, and because it's still in progress, I will present only the results of the combined chain, so the alpha and the beta chain together, for the effector and the regulatory T cell separately, and for the enriched and impoverished signature separately. And here you have this heat map that represents the area under the curve of each prediction. You have in column the different type of learner, and in row you have the different conditions. First, the iteration. Here you have also the disease. We added COVID-19 to have a positive control to evaluate if we are able to classify also when we separate the two cell subsets. We have the effector T cell and the regulatory T cells, and the interesting part is signature. So we have in green the enriched signature, and in red the impoverished signature. So we identify two type of clusters. The first one, that group poor prediction of autoimmune disease, mainly autoimmune disease, and this is a prediction with the impoverished signature. The second cluster, that's group majority of COVID positive control patients, and we also have the signature from enriched autoimmune disease patient. So to explain these two type of results, so we have good prediction with COVID-19, probably due to the strong and recent immune response, and maybe we are much more in this case, so that's why we have a higher prediction score. We have heterogeneity for the autoimmune disease with some iterations that with a good prediction. And maybe for the autoimmune disease is due to the chronic response and the heterogeneity within all groups due to the disease duration, the different treatment, and the age of the patient. So we are mainly on this type of representation. But overall, we have this enriched TCR signature from effector T cell and regulatory T cell, both, that allow us to classify the COVID and the autoimmune disease from healthy volunteers. The second part is to compare this signature. So here we focus only on the autoimmune disease, and we will create the final signature with the full dataset by group. And we obtain again the enriched and the impoverished signature, and we will compare the sharing between them. So here on the top of this vein, you have the impoverished signature and the enriched signature. Overall, we observe that we don't have a high sharing between them. And when we perform a clustering based on this signature, so here you have the result of the clustering for the effector T cell on the left, for the regulatory T cell on the right. Each column represents a patient, each row it's a signature. When the signature is present in the dataset, it's colored in yellow and absent in black. And we observe for both cell subsets that all signature allow us to have a good clustering purity with open 85 and 88 for the regulatory T cell. So overall, we observe that all feature selection and all signature are private for each autoimmune disease and each cell subset. Finally, we would like to characterize this autoimmune disease with external dataset because they allow us to predict and to cluster different patients due to their pathology, but we don't know if this signature that we observe in blood are involved in the immune response, because we don't know what we observe in blood is overlapping with the organ. So for that, we compare our signature with external dataset. And because our signature are composed mainly by alpha chain, we have a low proportion of beta chain in our signature, and we have to evaluate why we have this difference. The beta chain represents around 10% to 40% of the signature, and the external dataset are only on beta chain. We need to increase the overlap to increase the information. We use the Levenshtein distance as a reflection of the TCR similarity. So for each of the signature, we apply the Levenshtein distance and also the perfect match between the CDR3 with the external dataset, and we will observe the sharing in the dataset of our signature. So the first result is for the rheumatoid arthritis patients. So we extract the dataset from Albany paper. So they have a cohort of six juvenile idiopathic arthritis from blood and synovial fluid for the effector T cell, and also that they characterize the circulating pathogenic lymphocyte in the blood, and as I say, for the beta chain. And so this is not rheumatoid arthritis patient, but in the paper they show that these circulating pathogenic lymphocytes are also found in rheumatoid arthritis patients that are expanded in rheumatoid arthritis patients compared to the healthy volunteers, and this proportion increase with the disease activity. And when we compare this or signature in this dataset extract from this paper, we observe two significant differences. So here we observe the proportion of our signature Levenshtein distance, the proportion by patient. So for the blood in yellow, for the circulating pathogenic lymphocyte in orange, and in red for the synovial fluid. And we observe first that our enriched TF signature is significantly enriched in the circulating pathogenic lymphocyte compared to the blood. And this confirms the pathogenicity of our enriched TF-CD3 that's composed our signature. The second point is our EMPO-rich Treg signature is significantly more found in the synovial fluid, in the effector T cell in the synovial fluid. So our Tregs, EMPO-rich CD3 that compose our signature in the blood of our rheumatoid arthritis patients are detected in the effector T cells in the inflammation site. So this Tregs EMPO-rich could be a protective Tregs that are EMPO-rich in the patient. The second validation is now extracted from Todd Brusco paper for the T1D patients. So they have 18 T1D patients, 9 healthy volunteers. They have the pancreatic lymph node for the effector and the regulatory T cell. And again, we also here found a difference between the healthy volunteers and the type 1 diabetes patient for the enriched TF signature. This signature enriched in our patient is more present in Tregs of the pancreatic lymph node from the T1D patients compared to the healthy volunteers. So this result suggests that our effector enriched signature is autoreactive. And we also found that in the patient, we have a high proportion of Tregs in the pancreatic lymph node from type 1 diabetes patient. They are in the seat of the inflammation, but they could be not effective due to a lack of interleukin-2. And finally, to evaluate the autoreactivity of this enriched effector T cell signature in rheumatoid arthritis patient and in type 1 diabetes patient, we perform a modeling of the selection process. We used a Sonia algorithm proposed by Alexandre Balzac and Thierry Mora and collaborators that allow us to create a model based on our datasets, a selection model to identify specific sequence features, characteristic of functional lymphocyte repertoire. So for each of our groups, so for the healthy volunteers, type 1 diabetes patient and rheumatoid arthritis patient, we split the cell subset. So we create a model by disease and by cell subset. With this model, we compute the probability of selection based on this model of our signature. And we observe first, and this data is not shown here, but we observe that we have a significant higher selection probability for our enriched effector T cell signature for RA and T1D patient with the disease model, whatever the cell subset. So for TF and for Tregs when we compare to the healthy volunteers. So we select these two different significant difference values, and we compute fold change to this fold change between these two probability of selection, and this is represented here. So here you have the fold change between the healthy volunteers and the diseased patients for the TF model of RA patient, the same here for the Tregs RA patient, and also here for the T1D patient. And we could observe here that we have a significant difference to have with a higher, we have a significant difference with a higher difference within the Tregs model. So these results for each of these diseases, these results characterize that the model, the Tregs model indicates a higher probability of this enriched TF cells to be selected as Tregs. So that tend to confirm these self-reactive properties. And we could hypothesize that we observe here a bias in the selection process, so self-reactive T cell pass negative selection, but also a proposition of Todd Brusco in this paper that is a conversion from regulatory T cells to effector T cells, because the Treg lineage and stability and plasticity have been suggested to contribute to autoimmunity via the expansion of autoreactive CD4 T cell from a regulatory parent clone. Finally, and to conclude, overall this presentation that is still in progress, this work is still in progress, we provide a new proof of concept that TCR repertoire could be used as a biomarker in the blood. We are able to predict the outcome of a COVID patient with a high accuracy. And when we sort Tregs and TF cells, we are also able to have a good prediction for the infectious disease, lowest than when we observe the total T cell, probably due to the presence of CD8 T cell in the adaptive data set. For our autoimmune disease prediction, we have accuracy that is lowest, that is lower than COVID patients, probably due to the difference between the response for an infection and autoimmunity. And we have to add a new control for this part with a random signature, as I presented for the COVID from adaptive results. And finally, so by comparing blood and tissue as well as different subsets, we suggest that these enriched TF signatures in both disease have pathogenic properties because they can be identified in the inflammation site. This enriched TF signature could escape the thymic selection because they are shared with also Tregs present in the inflammation site. And this enriched TF signature may originate from Tregs and convert into a pathogenic effector lymphocyte. Finally, thank you to David Klasman Laboratory and to the TCR team led by Anca-Anita Mariotti-Ferrandis, and also to the Human Immunology team led by Michel Rosenwag and all the collaborators and sequencing facility. Thank you.
Thank you very much, Pierre. Very, very interesting. Well, first of all, people are welcome to ask questions in the chat or here. I have a comment, a very silly comment, but it took me a while to understand that AID is actually autoimmune disease. So in this audience, you know, AID has some different connotations. So maybe you can choose a different abbreviation. I'm not sure if it is related to you, but someone asked here, what are the features of the TCR repertoires and how do you process summarize these features for your prediction model? I'm not sure if it was directed to you or to Phil, but maybe to you. Can you comment on this?
When you say the feature, you mean which region of the TCR?
Yes, indeed, it was to you. Maybe you can unmute yourself, Jonathan, and ask.
So for our prediction, we select the CDR3 region because we think that because this is the region that interact with the antigen. For this, for our patient, we don't have the HLA, so without the V and the gene, the G genes that interact with the HLA, we propose this strategy to just focus on the CDR3 that
right, but I mean, like, sorry, I mean, is it like a one-hot encoding of your CDR sequences, or is it, you know, like, are you passing it through like a Word2Vec? Like, what do you mean by just CDR3 sequences? That's pretty loose. I'm not gonna lie. And you say signatures a lot, but I mean, like, what is a signature? Because I still, if I'm honest, I still didn't get that from the talk.
Sorry, I don't understand.
Maybe I can rephrase it. I mean, when you say CDR3 sequences and use them to the classification, do you mean like the sequence of amino acids that is probably shared between individuals, or do you encode it somehow with one hot?
No, we just select the CDR3 in terms of amino acid, and we just select this sequence, whatever the clonotype, whatever the V or the G, and we just use this information.
So it is like public CDR3s that are used for this prediction.
That yes, this is because I don't present the feature selection, but we select public clones that are enriched in one group compared to the other in terms of amino acid.
Okay, so maybe I'll have a follow-up question on this. If you then take all of these features, these CDR sequences, can you find anything in common? Do you have like any motifs that are shared between these public CDRs?
We try, we are, that's why I say we are still in progress, because we try to find some motif between all of this group of CDR3 in terms of exact sharing. I show in the vein that we don't have a high sharing between TF or Tregs in which or other thing. But we try with algorithm like dipTCA or glyph to find some motif in this CDR3. So this is the next step to characterize this CDR3. Yes.
Okay, thank you very much. Any more questions or comments? Okay, I have a last nasty one. When do you expect these sequences, this great data that you produce, to be accessible by others? I told you it was nasty, and Bernita.
When we will publish the data and the results. So this paper, this work, we are now working on summarizing everything on the paper, but as Pierre said, there is still some parts that we need to confirm.
But meanwhile, okay, sorry, but meanwhile, we're open for collaboration. So, data is available for those who would like to work with us on that and for collaboration.
Challenge accepted, David. Thank you very much. All right, so I think we've got to the half hour. So I would like to thank again our two speakers. It was extremely interesting. And invite you all to our next seminar, which will happen in April, the last week of April. So thank you very much.
Thank you, everyone.